This is lecture 10. We're back now to talking about this thought that in some cases a blood test may indicate some problem with kidney function. And let's take a specific example. Let's say, for example, that a patient has a high urea level in their blood plasma. And the question is, what does that indicate? Well, quite possibly, it indicates that not enough urea is being filtered out of the blood in the glomeruli. So again, high blood urea could indicate that quite possibly you have this excess of urea in the blood because not enough urea is being filtered out of the blood. And the next question is, why would this be? And the answer is, well, because quite possibly the GFR, the glomerular filtration rate, the rate at which this filtrate is forming and the rate at which this urea is being removed from the blood, quite possibly then, because of some problem with kidney function, this rate is too low. And I'll give everyone just a minute here to jot down what you want to in your notes. But again, the idea, a blood test in some cases may indicate a problem with kidney function. As an example, too much urea, too high of a concentration of urea in the blood. Urea, again, is a waste product that we want to filter out of the blood. And so again, if there's too high a concentration of urea in the blood, it may indicate a problem with glomerular filtration. Not enough filtration is going on. The rate of filtration, the GFR, is too low. And again, this could quite possibly be because of or this could indicate a problem with kidney function. At this point, I want to refer to a figure that's not labeled in your lecture outline. So before we get into, on your outline, talking about control of filtration rate, let's take just a minute and look on page 782 at figure 2017. And notice we're looking here now at two different bars showing on the left glomerular filtrate and on the right urine, and we're looking at over a 24-hour period how much glomerular filtrate, what volume of glomerular filtrate is typically formed. That big bar on the left, and then to the right, also within a 24-hour period, we're looking at how much urine formation and release is typical. And notice then glomerular filtrate production over 24 hours, on average about 180 liters. Just think about what that means. Within all of these small glomeruli in the kidneys, we have all of this filtrate forming, and again, this filtrate forming at the rate of 100 liters every 24 hours. But again, we only lose through urination about 2.5 liters of urine. And of course, what this means is that most of that fluid that's been formed by filtration, most of that fluid is going to be reabsorbed by the body and not lost as urine. And again, I'll give everyone just a minute to let that sink in and jot down whatever you would like to here in your notes. Back to your chapter outline now, and we're to the point now of talking about the control of filtration rate. And notice two different types of control going on here. And so number one, glomerular filtration rate remains relatively constant through a process called autoregulation. Your book doesn't talk any more about that process, and we're not going to talk in any more detail about that process. But basically, filtration rate stays pretty much the same with some fine tuning. Number two then gets into that process of fine-tuning filtration rate. So number two, back to your lecture outline again, the sympathetic division of the autonomic nervous system allows more precise control of glomerular filtration rate. And notice now we're looking on page 784 at figure 2018. And notice in that figure, there are upper left an A portion, upper right a B portion, lower left a C portion, and lower right a D portion. Notice also what we're looking at here is, in each case, a glomerulus. And notice in each case we're looking at the afferent arteriole with the blood coming into the glomerulus, so the upper left, and that vessel that's shown in each of these four figures. And then notice also we're looking at the efferent arteriole, so to the right part, after the blood has gone through the glomerulus, we're looking at what's going on with this efferent arteriole that carries blood away from the glomerulus. To your handout then, to your lecture outline, and so first of all, looking at the A portion of the diagram, vasoconstriction of the afferent arteriole decreases net filtration pressure. And of course, anytime we decrease net filtration pressure, as we've talked about, we're decreasing GFR. We're decreasing the rate of glomerular filtration. Higher pressure, higher rate of filtration, lower pressure, lower rate of filtration. So let's take a look now again at that A portion of the diagram and think about this one for just a minute. If we vasoconstrict the afferent arteriole, if we make the diameter of the afferent arteriole smaller, this is going to cut down on how much blood enters the glomerulus. Not enough can squeeze through and into the glomerulus if we've constricted the diameter of that afferent arteriole. And without as much blood in the glomerulus, that means the blood pressure is not going to be as high. And with that lower blood pressure, we're not going to be forcing as much filtrate out of the glomerulus. So again, vasoconstriction of the afferent arteriole decreases net filtration pressure, because when you decrease that diameter of the afferent arteriole, less blood gets into the glomerulus. This results in lower blood pressure, lower glomerular hydrostatic pressure, if you will, forcing blood out of the glomerulus, and so decreasing net filtration pressure and decreasing glomerular filtration rate. The B part of the diagram then, so now looking at vasoconstricting the efferent arteriole. Notice for each of these figures, there are arrows to the outside of the vessel showing vasoconstriction and where it's occurring. So again, B now, those two arrows in the upper right, just to the outside of the efferent arteriole, indicate we're decreasing, we're vasoconstricting the diameter of the efferent arteriole. This will, to your handout then, vasoconstriction of the efferent arteriole increases net filtration pressure. And if you think about it, that simply means more blood is backing up within the glomerulus. Higher blood volume means higher blood pressure, means increase in net filtration pressure. The C part of the diagram, so lower left, simultaneous vasoconstriction of both the afferent and the efferent arterioles. So find those two vessels, and notice now in the C part, in the lower left part of the diagram, we're seeing arrows indicating simultaneous vasoconstriction of both vessels. And back to your handout then, back to your lecture outline, this slightly decreases net filtration pressure, but not as much as it would if just the afferent arteriole was constricting. And then finally, for the D part of the diagram, so lower right, vasodilation now of the afferent arteriole only increases net filtration pressure. Notice the arrows again now showing, now kind of leading away from, in other words, indicating an increase in diameter of this afferent arteriole, bringing blood into the glomerulus. And if that's the only thing that's going on, if we're not messing with the efferent arteriole, then if you think about it, vasodilating that afferent arteriole means more blood is able to come into the glomerulus. More blood, again, means higher blood pressure. That higher pressure then forces more plasma out of the glomerulus, and so increases net filtration pressure. This is a good point to stop here. We'll come back next lecture and pick up beginning with the renin-angiotensin system and talk just a little bit more about this. So before you listen to the next lecture, take a minute and just recall what you know about that renin-angiotensin system. And again, we will go into that and review it a bit, and then also go into a bit more detail.